Hello, I'm Chris Rogers. I'm a physiatrist in San Diego, California. I'd like to thank Steve Sampson and the OrthoBiologic Institute team for inviting me back to this year's conference. I appreciate that very much. I will be presenting a clinical trial that was approved by the FDA on the safety and efficacy of an autologous adipose-derived stromal vascular fraction product for the treatment of knee osteoarthritis. These are my disclosures. I'd like to remind everybody that stromal vascular fraction, which is derived from adipose tissue, is a heterogeneous collection of cells and varies from one individual to the next, both in terms of the types of cells, the proportion of cells, and the number of cells. Of course, the most popular cell is the adipose-derived medicinal signaling cell, but there are many other cells, including progenitors, such as parasites and endothelial progenitor cells, and mature immune cells, such as macrophages, T cells, B cells, and monocytes. Adipose tissue can be processed by a number of different means, shown here, everything from unprocessed lipoaspirate on the left to collagenase enzyme-digested adipose tissue stromal vascular fraction on the right. Then there are intermediate steps where you can rinse, wash, and resize the adipose tissue. But it's important to note that these are all different in terms of the content of cells, with there being more concentration of stromal stem cells in the stromal vascular fraction. For those of you not familiar with adipose tissue, I use this Lucky Charms analogy where we know it is magically delicious because it has these colorful marshmallow bits, which represent the cells in the stromal vascular fraction. And as you further process the bowl of cereal on the left, you can further concentrate those marshmallow bits. Cultured adipose stem cells would be simply taking those blue diamonds and culturing them to get a nearly 100% stem cell product. There have been a number of human clinical trials on adipose-derived orthobiologics, but on MFAT as well as SVF and ASC. Many of these have been performed outside of the United States, and only one has been performed with FDA approval here in the US. And this is a long-standing challenge in orthobiologics in that we rarely characterize what is in our PRP, our BMAC, our adipose tissue grafts. And that's what drew me to a GMP processed product where we can actually know what's in our soup. And so the adipose tissue is processed in an FDA-inspected facility where we can then have the identity, strength, quality, and purity of the product well characterized. And so we can do cell counts, we can look at flow cytometry to identify cells, we can measure viability, we can test for sterility, mycoplasma, endotoxin testing, and then package the cell in a sterile bottle and ship frozen to the physician's clinic. Of course, this succeeds minimal manipulation, and the FDA states that SVF is a drug, and therefore it has to go through the regulatory pathway for phase 1, phase 2, phase 3 studies before applying for a BLA and allowing you to distribute the drug in the marketplace. So you can go on clinicaltrials.gov to look at our protocol for our study, which is an FDA-approved study. We had 6 physiatrists as investigating physicians. We had 3 orthopedic surgeons and 1 interventional radiologist. Many of these folks are speakers at prior TOBI events. And I will also mention Dr. Harmon and Mike Dale, who founded a company called VetStem, which has more than 20 years experience treating animal patients with stromal vascular fraction. And this data allowed us to then get FDA approval to conduct our human clinical trial. The inclusion criteria included patients age 18 to 80 years of age, any gender, with history of osteoarthritis and weekly pain for at least 3 months, radiographic evidence of 2 to 4 grade osteoarthritis, and had failed conservative care for at least 6 weeks. Many of these patients, of course, had pain for many, many years, and then they were otherwise healthy. We excluded them if they had contralateral knee osteoarthritis, Kellgren grade greater than 2, or had had injections into the treatment knee, or if they had had medical conditions that precluded them from undergoing treatment. Our primary outcome, of course, was safety, and this was assessed by history, physical examination, and laboratory tests with CBC, metabolic panel, and urinalysis, as well as observations after treatment. And patients were able to report adverse events for up to 1 year. We did a preliminary look at efficacy using the COOS, which is a 42-item survey with 5 subscales, which is commonly used for knee osteoarthritis, and the WOMAC, which is calculated from the KOOS, which is a 24-item survey with 3 subscales. We also looked at patient satisfaction at 1 year. So we used a standard tumescent anesthesia to provide adipose collection, and then adipose tissue was harvested from either the flank or the abdomen using a typical cannula and syringe with a target volume of about 100 to 150 mLs of lipoaspirate. The tissue was decanted of excess waste fluid and then placed in a validated shipping system where it was FedExed overnight to the GMP facility for processing. And again, here you see the GMP facility where they're processing the adipose tissue to manufacture the SVF product. It is then sent back to the clinic in a liquid nitrogen charged dewar to keep the cells frozen. They're then thawed and injected into the knee with a standard ultrasound-guided injection technique. The injection was performed into the suprapatellar recess, again, using standard technique. And then our investigators validated this technique. All right. So we had lot release criteria of a minimum of 2 million viable cells, nucleated viable cells. The dose ranged from 2 to 8 million, depending on the individual, with an average viable cell dose of 4 million cells, of which about 20% of these were the ASC, and then the others represented down below where you see there. We had 38 patients enrolled, but 29 actually received a single injection of the study drug. These patients had a mean age of almost 66 years of age, a KL grade of 3, and a BMI that put them in the overweight category. I'll notice that with a KL grade of almost 3, that's a little bit more severe than other studies that have been published on the subject. And we did not lose any patients to follow up at 12 months. We looked at safety. There were primarily grade 1 and grade 2 adverse events, of which only 6 were deemed to be related to the treatment. The others were unrelated. There were no serious adverse events related to the treatment, although we did have 1 patient fall off their horse and break their hip, but not related to the treatment. And there were no clinically significant changes in laboratory values, BMI, or vital signs. We looked at efficacy with the KOOS. The mean at baseline was almost 53. The response rate at 6 weeks was 82%, and we define response as exceeding the minimal important change as recommended by the developers of the KOOS survey. At 12 weeks, we have preservation of the response rate at 79.3%, with a mean improvement of 43.4% in the KOOS. And here you see the subscales represented, with the gray line representing baseline, the yellow, the minimal change to be an important clinical change, and then at 6 and 12 weeks, exceeding that MIC. And the WOMAC here, the baseline is the blue line. You see a reduction in the WOMAC of about 44% at 12-week follow-up in each of those subscales. And this is very comparable to prior studies that were published on SVF or cultured adipose stem cells. This is a meta-analysis published in Cells journal this year, where at 3 months, the average of all the studies was about 42% improvement in the WOMAC. So again, we saw 44%, so it's very in line with the prior published literature. We have 12-month data where we look at patient satisfaction, where the patients submit a form to the sponsor, and we have about 74% patients reporting that they're very satisfied or satisfied. Of course, we don't have all the data in yet, but we should by the end of this month. So in conclusion, we have a very good safety profile, no serious adverse events, clinical efficacy at 3-month follow-up. We are almost done with our 12-month follow-up, and I'll be publishing that data shortly. And then we're going to start a phase 2 randomized clinical trial before the end of this year. I'd like to thank the folks at Personalized Stem Cell, as well as our investigational physicians and their support teams. And if you have any questions at all, please email me. I'll be happy to give you a copy of the publication once it's completed. Thank you very much.